Krieger. The date is Wednesday, February 22nd, 2023. And my name is Rachel Krieger. I'll be presenting on the steroid hormones, as well as protein hormones and intracellular proteins. We'll first begin with the steroid hormones. So each of these cells that are intended to create or synthesize these hormones have the DNA and molecular kind of makeup to create these steroid hormones. They're able to process cholesterol, which we can see the molecular makeup of cholesterol here. We can see the hydroxyl group, the steroid nucleus, and the hydrocarbon side chain. And the cells are able to take that and convert it into pregnenolone, which we see the same hydroxyl group and steroid nucleus. However, we do see a carbonyl group exchange for the hydrocarbon side chain. And all those processes are taking place in the cell. So first we will have the cholesterol, the precursor to the steroid hormones, go into the cholesterol is first synthesized in the endoplasmic reticulum and then is able to move to the Golgi apparatus. Then in the Golgi apparatus, it is processed and created kind of in a way that can make it ready for the mitochondria to synthesize the final product, which would be the steroid hormones. And so the mitochondria contains the cholesterol side chain cleavage enzyme P450-SCC. And with that, the cells are able to convert cholesterol to pregnenolone, as I showed down here, with the aid of this enzyme, as well as other steroidogenic enzymes like hydroxysteroid dehydrogenases, as well as many more. The rate-limiting enzyme CYP11A aids in the cleavage of the side chain of cholesterol, as we can see here, and is able to convert that to pregnenolone with the aid of these, as well as other aforementioned ones. The endoplasmic reticulum then is able to create more enzymatic reactions than that produces the final steroid hormone, such as cortisol, testosterone, or estrogen. In their final form, these steroid hormones are able to leave the cell with no external aid because they are lipid soluble. So they're able to pass through phospholipid bilayer just through simple diffusion. And since the concentration is high within the cell compared to the outside of the cell, this simple diffusion is taking place because the concentration is higher within the cell as compared to the outside of the cell, and therefore no energy is expended by the cell itself. Once outside the cell, the hormone is able to bind to transport proteins that are outside the cell, and they aid in reaching the target cell and aiding the steroid hormone in reaching that target cell that it was intended for. I got my information for this. We can protein hormones. Protein hormones begin when mRNA transcribes the DNA information located in the nucleus, and it creates that for protein synthesis. The ribosomes located in the cytoplasm or on the rough endoplasmic reticulum are able to translate the genetic code into amino acids or then to cytoplasm within the cell, and they're joined together through long chains by the ribosomes. These long chains, now known as polypeptides, can now be released into the extracellular matrix through facilitated diffusion. The exact protein type in which helps these cells help these polypeptides exit the cell is through a carrier protein. The way that carrier proteins work is they are able to kind of absorb the polypeptide, and then through their lipid-soluble outer membrane, they're able to carry the non-soluble lipid in through the phospholipid bilayer, and then they release the polypeptide into the external cell area. And I got my information for that with Nunari in 1991. Next, we can talk about intracellular proteins, which are made by ribosomes in the cytoplasm in their final form. In a similar manner as the protein hormones were made, the intracellular hormones are made beginning in the DNA. The DNA is then transcribed into mRNA, and mRNA is then translated by the ribosomes within the cytoplasm and not on the rough endoplasmic reticulum. Then, once they are released as amino acids and formed in the polypeptide chains and created as proteins, once they are functional, they are able to go wherever they are needed within the cell. I got my information for that from Yay 2010. And these are also my references. Thank you.